Hello everyone, my name is Bruna and I am a graduate student at the University of Melbourne. In this poster, I will share about our work on predicting conformational B-cell epitopes using graph-based signatures. My supervisors are Dr. David Asher and Dr. Douglas Pires. In the background, to start, I would like to briefly remember a biological concept. B cells are an essential part of the immune system that provide long-term protection against pathogens or antigens due to the antibodies. And how antibodies identify an antigen and eliminates it? Binding to them. Antibodies connect to a fragment of the antigen called epitope. And most B-cell epitopes are of the conformational type. They are also called discontinuous because they are composed of residues that may be far apart in the sequence, but spatially close in the protein structure. In general speaking, epitopes represent only 4% of the entire antigen structure. And this information makes us realize the big computational challenge dealing with a high imbalance data. In this way, epitope identification could lead to a higher disease response in the design of vaccines and also in the immunotherapy process. And those are our key motivation in this work. So, Epitope 3D is a tool that uses a machine learning model combined with structural signatures to predict conformational epitopes. And our method comprises 3 steps. First is the curation of the dataset, the feature extraction and selection called feature engineering, and the machine learning process. In the data curation, we aim to identify unbound antigen structures, but based on experimental antibody-antigen complexes, and these are all extracted from the Protein Data Bank, the PDB. The idea here is to reduce the false negative annotation, so different epitope regions from related antigens are extracted and aggregated in one unique antigen, based on sequence and also structure similarity. After that, we extract characteristics of both epitope and non-epitope residues. And we adapt the concept of graph-based signatures to represent here the geometry and the chemical composition of the environment surrounding an epitope. So the graph involves modeling atoms as nodes and their interaction as edges, basically. Also labeling these atoms according to their physicochemical properties. It's important to highlight that several other features were assessed in this step as well. And moving to the last stage of our method, we have the machine learning process. The machine learning includes a qualitative data analysis of the selected features, the training and model optimization using different supervised learning algorithms, and they were all evaluated by 5, 10, and 20 cross-validation technique. Moving to the results, to conclude, I would like to share the summary of our results with you. We have curated the larger, non-redundant set to date, achieving 245 antigen structures. From this set, we are releasing 45 as an independent set to evaluate the performance of other available methods as well. Epitope 3D achieved a good performance on cross-validation with Matthew's correlation coefficient of 0.55. And taking the benchmark dataset, the 45, as I said, to compare our model with other well-known B-cell epitope prediction like CIPA, Discotope, ELIPRO, and BPPRED, our predictor outperformed all of them, reaching a MCC of 0.45. even with a highly imbalanced set here. Finally, our predictor is already available as a friendly web server. You can access it using the QR code provided at the bottom here. And feel free to contact us there or also visiting our Twitter accounts as well. Thank you all.